Okay, so now you learn about cloning and how we can make recombinant DNA molecules. So let's talk a little more about other techniques that we have in biotechnology. So genomics, if you remember, is like the entire genomes of a macroorganism and so including all the genes, sequences, and also organizations. So it has a very important role in medical advances, also for finding new antibiotics, vaccines, and treatment. So that's very important to have like the knowledge of all the genetic information of the organism. So the more you know about that organism and their genes, it's easier to do the treatment and uh, the making medicine and like the drugs against them. So in genomic library, we're gonna extract all the DNA from the organism, and then using the restriction enzymes, if you remember that they act as like a molecular scissors. So we cut the DNA fragment in a small pieces using like one or two restriction enzymes, and then we cut the plasmid with the same restriction enzymes, and then with the help of the ligase we can insert these DNA fragments into the plasmid, and then we can transform the bacteria to the transform the recombinant DNA to the bacteria, and then so the bacteria can replicate and it can produce like colonies of that clones that we have. The interesting thing about this technique is that each of these colonies can carry like a different kinds of genes or different DNA fragments. So and then based on our need we can target a specific gene, so we can pick that specific colony of the bacteria, and then we can grow it and extract that specific like a fragment of the DNA or the genes of interest from that colony. So also we can make recombinant phage DNA. So if you remember, bacteriophages are the viruses that can infect bacteria. So they can use the phage particles in order to make like a recombinant DNA. So and they use it especially for bigger DNA, so bigger because you can clone and insert like a bigger fragments to the phages in compare with the plasmid from the bacteria, and the phages particle can hold a much larger insert of that foreign DNA compared with a plasmid vector. So what they're gonna do is pretty much the same. They're gonna digest the cellular genomic or DNA with the restriction enzyme, and then they use the same restriction enzyme to cut the phage genome. Then they're gonna build the recombinant phages. Then these phages can infect like bacteria such as E. coli. So while they infect it, so they can move this recombinant phage DNA to the bacteria. So and depends on some phages are lysogenic, some of them are lytic, so depends on, it varies. For example, if it's lytic, so it's gonna kill the bacteria, that it makes like some clear areas in the culture that they call them plaques. So plaques are the area of the bacterial culture that the phage actually lysed and killed the bacteria, and you can see it like as clear area on the petri dish. So and so each plaque can represent a unique recombinant DNA, and you can screen it based on the genes of interest, and then you can do further study on that genes that you are looking for. So another thing we can do, we can make like the cDNA from the messenger RNA using reverse transcriptase. So sometimes we don't need to have like all the genetic information of that organism. We just want to focus on the messenger RNA or on the genes that can be translated and make proteins. So for this reason, instead of extracting the whole genomic DNA from the macroorganism, we're gonna just extract the RNA from that macroorganism, and then because RNA is not a very stable macromolecule, so we need to convert it and we can change it to DNA, or they call it cDNA, complementary DNA. So for converting RNA to cDNA, we have to use an enzyme that they call it reverse transcriptase. If you remember, transcription means making messenger RNA from DNA. So reverse transcription or reverse transcriptase, so these are the enzymes that they can make DNA from RNA. And actually they extracted this reverse transcriptase enzyme from retroviruses, that they are RNA viruses, but they have this enzyme to be able to convert the RNA genetic information to DNA. So after the reverse transcription, so we're gonna have the DNA or cDNA, and then they're gonna use restriction enzymes to cut the cDNA and the plasmid, and then they ligate it together and they transform it to the bacteria. And exactly same as before, each of these bacteria replicates, they can produce like a unique colonies that carries like a specific fragment or genes, and then you can do the screening to find like the genes of your interest that you can do further study. So they call this cDNA library, and the main difference is that with the genomic library, that our main focus here is studying the proteins, not the full genome sequence that happens in the genomic library. Okay, so now we're going to talk about transfection. This is a process that we're going to introduce recombinant DNA molecule into eukaryotic cells. It's much harder to introduce the recombinant DNA into eukaryotic cells in compare with prokaryotic cells, because if you remember, we talked about transformation, so we easily can use some chemicals or can use electricity to increase the capability of the cell membrane in bacteria to accept or to get the recombinant DNA, but in eukaryotic cells it's much harder to enter the recombinant DNA into the cytoplasm. So there are several techniques that they use it in order to transfer this recombinant DNA into the plant and animal cells in order to make genetic engineering plants or animals. So one of the technique they use, they call it electroporation. This is a technique that they use it very commonly in order to introduce the recombinant DNA in plant and animal cells. So they can use like a brief electric shock or pulses that can create temporary pores, very small pores in the cell membrane, and then it's gonna let the DNA to enter into the cell and cytoplasm. So another technique that they will use in eukaryotic cells is microinjection, especially in animal cells. So they can use very small, like microinjection needles, and then they can transfer the DNA into the eukaryotic cells, and so they can. This is how they can. The needle can penetrate inside the cell or in the nucleus, and then they can inject the recombinant DNA into the cell. They also use gene gun, so especially in the plant cells, so they can use because plant cells, they have cell walls, and it's much more harder to introduce the recombinant DNA into the plant cells. When they use this like a gene gun, is gonna remove the cell wall with the help of some enzymes, and then by using these guns, so it has like a helium pressure, and it can shoot the recombinant DNA into the cell, and so especially for plant cells making transgenic plants. So this is a very common technique that they use, and then the cell can recover, and they can use it for generation of the recombinant plants. Another important technique that they use it, they call it TI plasmid. So tumor-inducing plasmid. This is a plasmid that we have it in a bacterium. They call it Agrobacterium tumefaciens. So this bacteria can cause tumor in plant. So with this TI plasmid that has, so it's like a cancer for plant, and so they modify this TI plasmid, so they uh, uh, remove those like pathogenetic genes from the plasmid, and then they introduce or they insert that gene of interest, so they can clone it, or they can clone the gene of interest into the TI plasmid, and then they can introduce the TI plasmid into the plant cells. So the good thing, or the interesting thing, is that this TI plasmid has this capability to become a part of the genomic DNA, so it can get integrated with the genomic DNA, and then it can help to produce transgenic plants. So it's another common technique that they use it for making transgenic plants. So now hopefully you have a better idea about how we can introduce the recombinant DNA into eukaryotic cells. So the next part, we're going to talk about how we can visualize and characterize DNA, RNA, and protein. So one of the technique that you need to know, they call it DNA probe or DNA probing. This is a technique that we use it in order to isolate and identify specific DNA sequences by using DNA probes. So probes are a small fragment of the DNA that they usually label them with the radioactivity or fluorescence, and you know that the DNA has this characteristic that if you have like a single strand DNA can attach to the complementary strand of the like imagine target gene of the interest. So the first thing they're gonna do, they isolate the DNA, imagine from the blood or urine sample, so from the body fluid. Then they're gonna denature it to make sure that these the hydrogen bonds between the nucleotides gonna break down, and then you have a singular strand. So then we can purchase, or you can make it too, but they usually purchase these probes. So these are like imagine we want to find that specific infection in the blood, so we want to see that if this bacteria ex exist in the blood. So then they purchase that DNA probe, which is already labeled with radioactivity, but usually they use fluorescent, which is much more safer. So then if this DNA probe can match or is complementary to the sequence of that bacteria, so it's going to attach to it. There are several steps of washing, and then if the DNA probe bind with the gene of interest, it will show us that that gene or that bacteria present in that sample. So it can be used for diagnostic or it can be used for research. So very common technique that we use DNA probes. Another technique that we use to do the to visualize the DNA from our bacteria, fungi, from our infectious agent, we call it gel electrophoresis. And so and also especially after PCR, so that we're going to talk about it. So they use this technique to see that we get any amplification from that gene of interest or the DNA fragment in the PCR. So the agarose gel, this is a technique that they usually use it for visualization of the DNA or even RNA fragments. So and you can separate the DNA on a gel which is made of agarose, and agarose is like a products from the brown algae, and it's exactly like jello. So they mix the agarose with the buffer, they heat it, and after that they pour it on a tray, so and then it gets solidified, and then you can load your DNA sample to this like there are some holes in this gel, so you can load your DNA, and then they connect it to the electricity. You know the DNA has a negative charge because DNA has a phosphate group, so then the DNA based on their size and based on their charge are going to move inside the gel, and it's going to move toward the positive electrode, and usually smaller DNA can move faster than bigger DNA. So the DNA that you can see it like here, they are smaller than the DNA that are like move slower. And then we have something we call it DNA ladder or marker, that we know exactly there are several lanes, and we know exactly what is the size of the DNA fragment in the marker in of each of these lanes, and then by comparing our DNA with the marker or ladder, we can estimate the size of our DNA. And then usually they use a dye, so with the DNA, so they dye the DNA, and usually like a fluorescent dye, they call it cyber green. And then by using the illuminator, so the UV light, so you can visualize your DNA on the gel. So all these small clear or bright parts or fragments that you can see, or they call them band as well. So these are our DNA, and this is the ladder, so you can have ladder on both sides, and we know exactly what is the size of each fragment, and then we can compare it with the ladder, and then we can recognize or identify what is the size of our gene. For these techniques, probably if you haven't done these techniques, it's going to be a bit hard to understand them, but I highly recommend to watch the videos. I will provide some resources for you guys that you can watch the videos, and you can visualize and understand these techniques much better. So another technique that is old, but they still use it in especially forensics science lab. So they call it RFLP, or restriction fragment length polymorphism. So imagine there is a crime scene, and then there are some like blood or hair or nail in that crime scene. So we can use this technique in order to detect that if like this nail or blood belong to the murderer, or to that suspicious person that we are looking for, to see if it's a crime, if he or she did the crime or not. So they're gonna extract the DNA from that blood or that hair or that nail sample that they found it in the crime scene, and then they can digest or break down the DNA fragment by using restriction enzymes. And then they have like a gene bank of like all the criminals, or all the or the suspicious people that they think they were involved in this crime, and then they can compare the pattern of that DNA sample they found it in the crime scene with the pattern of that DNA from the gene bank that they have, and then they can identify that who is the person that did like this crime. So it's very common in forensic science to track like the criminals. Also they can use it for tracking the diseases, to see which part of the DNA has mutation, just by comparing the pattern after digesting with restriction enzymes with the healthy person. They can see that which fragment is absent or present in that mutant, and then they can identify like that genetic diseases. Very old, but still very useful technique that they use. Another technique, they call it southern blot. So this is a technique that we use the DNA fragment. So we separate the DNA fragment using the agarose gel electrophoresis that we just talked about. But because agarose gel is a very like a fragile, so it's like a jello, so it's not gonna last forever. So it's a very fragile structure. So they gonna, after they separate the DNA on the gel, they're gonna make like a sandwich. So they put like a big tray with the hybridization buffer. So because they're gonna hybridize the DNA with that, we're gonna use like a probe here as well. So then they put some filter paper, then they put our agarose gel, then they put some nylon membrane, and then they put some paper towel and wait to make sure to press the membrane completely to the gel. So after running the electricity, the DNA from the gel gonna transfer to this nylon membrane, so now it's much more durable than that agarose gel. So and then they use some probes that they are labeled with like fluorescence or maybe radioactivity. Imagine we are looking for a specific gene that is related to the cancer, or a gene that is related to the specific disease. Then that probe, if we have that fragment in our agarose gel or now in our nylon membrane, so that probe gonna attach and gonna match with that fragment or that gene, and then they call this like hybridization. And then because it has a fluorescence tag or label, we can visualize it and we can see that if our like gene of interest is there and in which part of the DNA or which fragment of the DNA located. So they call this southern blot because the scientist that discovered this technique was uh, uh, his last name was Southern. So then we have Northern blood. So there is no scientist with the name of Northern. They just play around with these names. So Northern blood, this is when you do the hybridization of the RNA. So instead of same procedure, instead of using DNA, we use RNA. And then we have Western blot. So Western blot, this is when we use for visualization and identification of proteins. So Southern blood related to DNA, Northern blood is related to the RNA, and Western blood is related to the protein. And there is no Eastern blood. Another important technique is macroarray. Macroarray is a very expensive and very uh, labor intense technique. So this is not a technique that you can use it like in labs, but they usually use it in like scientific researches or research labs. So in microarray, we're gonna use a very small chip. So think about like a chip in the computer, and then they're gonna attach like a very small fragments of, and you can see here, so they put a lot of like a small fragment of the DNA that they already labeled with like a fluorescence, and they usually use two kinds of color, red fluorescence and a green fluorescence. So these are like a little probes that if you want to just visualize it, so they have this like a little probes that they attach it like with and they have different colors uh, on this chip. And think about like millions of these small DNA fragments are there, and then they're gonna see that like and they usually use this technique for gene expression. So we are working with messenger RNA and cDNA or complementary DNA. So we want to see that, for example, you have a healthy patient and then you have a patient with cancer. So you want to see that which genes are related to the cancer. So you're going to extract the messenger RNA from both patients, and then you're going to make cDNA, and then you label those cDNAs with red or green fluorescence, and we already have these chips that they are ready, so you don't need to make them. So you purchase them, usually they make it for you, and then it has like a lot of small probes or DNA. So then, based on their complementary and this cDNA from the patient, the cancerous patient and the healthy patient is gonna match with these probes, and they're gonna attach to them. And then you put it in the machine gonna read it, and then it gives you like a color coding map, so you can see it here. So the ones that are just expressed, like the genes that are just expressed in the healthy person, you can see it as green color. The ones that they are expressed in the cancerous patient, you can see it as red color. And the genes that are expressed in both cancerous and healthy patients, you can see it as like a yellow color. So it's a very you need to do a lot of bioinformatic and computer work. So you have to do a lot of studies to be able to understand and interpret this data. And but they use it a lot in the cancer study to see that which genes are related or are expressed in the patient that has cancer in compare with the healthy person. And then they can use them, identify them, and then they can use them as markers to use it for identification of, for example, cancer in the future. Very interesting and modern technique, but as I mentioned, very expensive, and it's not very common to use it like in normal microbiology labs. So it's more in scientific labs. All right, so I'm gonna stop here, and then I will continue with the polyacrylamide gel electrophoresis, or